Jacob, can you hear me?
Yep, I can hear you.
Great. I'm trying a different microphone, so we'll see if this works. I'm going to walk around the table and talk, and you see if you can hear me, okay? Can you
Sounds
hear me? Can you
Yep.
hear me?
Yep.
Can you hear me?
Yep.
Can you hear me?
Yep.
Can you hear me? Can
Yeah.
you hear me? Can you
Yep.
hear me? Were you able to
Yep.
hear me?
Yeah, the whole time.
Okay, then it works. Excellent.
Awesome.
500 easy. What, your microphone? She has microphone eyebrows. Yeah, for $600. Yeah, we have to laugh. Why? Wait, you spent. Why did it cost $600? $600 on her eyebrows. Yeah. It's art. It's art. Girl, you're so bougie. You're literally so bougie. Yeah, but $600 for a tattoo is ridiculous. It's a special kind of tattoo. Here's my thing. You've gone to a tattoo artist and said, I'd like this tattoo on my eyebrows. No, they can't do it because they have to do like mapping. Oh my God. All 6 do it. Right? It's a special kind of tattoo, Amanda. So bougie. It took 2 sessions. I've only had everything. Wait, did they do one eyebrow at a time or half of each? That'd be funny. That's what I asked. I said, did they do? They did half. They did up to here. That's not during a full payment or we go finish. I feel like you like my art. You look at John Hagen and I do a terrible job. Like, if you pay us more, I will fix it for you. Until like my art has to work. Yeah, no kidding. I'd be like, scratch that.
Not everyone's taking it, so don't flash it around, okay? So Jacob, because of your quarantine, are you able to take the test in the academic student connections, or are we going to have to make other arrangements?
I took it in academic student connections this morning,
And
so I
then
had an
we'll send it
appointment.
to me. Okay, then I'll get it. All right, terrific. Thank you.
No problem.
Hey, Meredith,
Hello.
can you hear me all right?
Yes, I can.
Okay, cool. We're going to start very soon, so.
All right.
Okay, people on Zoom, are the slides moving?
Yes.
Okay,
Yep.
terrific.
Good question. I'll probably go because I went on Zoom
Last Friday?
last Friday and then I didn't pay attention to class Monday and she called on me for something and I didn't know what it was. But I got 100% on the homework. So.
All right, sports fans, let's get started. So John, Jacob, were you guys surprised at the World Series result at all? So you've got the very high budget Dodgers, pretty low budget Tampa. I was pulling for Tampa. Yeah, same here. Kershaw has choked so many times in the playoffs. He outpitched himself this time. He pitched beautifully. He had movement like I've not seen him do before. Yeah. No, kudos to that guy. He pitched really well. So I was pulling for the Rays, so personally, but that's me. I will refrain from discussing the exam since there are people who haven't taken it yet. I'm giving it back. Just keep in mind there are people who have yet to take it. So please don't flash it around. And my word, that is hard to read. Boy, that's still hard to read. Maybe I'll have to renegotiate my orange background. All right, we looked at how muscles contract. Let's look now at the neural control of muscles. Muscles are controlled by upper motor neurons, but that control is also regulated by input from sensory structures as well. So the lower motor neurons, remember, they issue from the ventral part of the spinal cord. Their cell bodies are located in the ventral horn. The axons will issue from the anterior horn of the spinal cord. They'll then innervate a motor unit in a muscle. And that connection between it, that is the motor neuron, and the muscle is mediated by the motor end plate, so-called neuromuscular junction. Now, strangely enough, and this may surprise you, it surprises me every time I hear the number. Of your upper motor neurons, would you believe, only about 10% of them directly innervate lower motor neurons. Most of them innervate interneurons. Interneurons. What are interneurons? Can anyone tell me, perchance, what an interneuron is? Give her a high five for me. Yeah, well done. That's exactly what they are. They are intermediate neurons between, they usually communicate between either sensory and motor networks or between other motor networks. So your dorsal roots of the spinal nerves are sensory in nature. Remember, your sensory motor neurons are pseudounipolar neurons whose dendrites are modified into sensory structures. They then send their axons into the, so the cell body is, so the motor neuron cell bodies are in the spinal cord. The cell bodies of the sensory motor neurons are in the DRG. Corinne, what's DRG mean? Dorsal root ganglia. Everybody point to your dorsal root ganglia. Back there. Back there. Okay, some of you pointed here. I'm like, no, no, no. Yeah. Rem was pointing like this, so he was the closest. No. Yeah. Sensory feedback is going to tell the muscle how far it's stretched. Also, it's going to, proprioceptors are going to give information on position of the muscle, how much it's stretched, and what have you. That is going to influence the motor input to the muscle. So your sensory to motor neurons are somewhere in the vicinity of about 2 to 1. You have a lot more sensory information coming from the muscle than you have information to contract going to the muscle. So in a lumbar segment, you have this gee whiz number here, 375,000 cell bodies. That's a lot more than you can account for by the number of motor neurons. In fact, these neurons, most of them are serving as interneurons. They will conduct impulses up or down or across the spinal cord. We already talked about ascending, descending tracks. I don't want to belabor this point. Just to remind you, descending tracks come from the brain to the muscles. They're motor tracks. Ascending tracks come from the peripheral sensors to the central nervous system. They are sensory tracks. Commissural tracks are those that cross the midline to the other side. They're not necessarily the result of decussation. They're more usually the result of interneurons. So those are commissural tracks. If they cross the midline, they're commissural tracks. And interneurons are those neurons that will connect with either sensory neurons or motor neurons and then connect them to other circuits. They may connect with circuits going across, so they may connect with commissural tracks, or they may connect with ipsilateral tracks. Let's look at the sensory organs in the muscle itself, the so-called muscle spindle fibers. Muscle spindle organs, muscle spindle apparatus, muscle spindle fiber organs are all the same thing. So your muscle spindles are your proprioceptors in the muscle. They detect the position, they detect the movement of the muscles. They will initiate reflexes that prevent you from damaging the muscle by overstretching or overcontracting the muscle. They're scattered throughout the muscle, and they operate very much on an unconscious level. The tendon will also play a role in this type of control of the muscle. You have a different organ there. You have a so-called Golgi tendon organ in the tendon. Both of these are going to work to convey information about muscle position, degree of contraction, and what have you. The muscle spindle apparatus is in the muscle. The Golgi tendon organ is in the tendon. They're both sensory organs. So this is a light microscope picture of a cross-section of a muscle, skeletal muscle. Now you have two types of fibers in the muscle. You have the fibers that actually generate the force of muscle contraction. And these are these fibers that we've been studying. These are so-called extrafusal fibers. These are innervated by lower motor neurons, and the particular lower motor neurons that innervate them are called alpha motor neurons. In this cross section, you also have a muscle spindle fiber organ, muscle spindle apparatus, whatever suits your nomenclature fancy. This also contains fibers. These are called intrafusal fibers. Intrafusal fibers. Intrafusal fibers. Now, these intrafusal fibers, they contain actin and myosin and so on and so forth. They don't, however, contribute any strength to the muscle. And it turns out that the contractile proteins don't extend through the entire length of the intrafusal muscle. They're only present in certain portions of it. So these intrafusal fibers, they're going to contract with the extrafusal fibers, and then sensory neurons are going to tell the nervous system about how much contracting this muscle is doing based on a sample, if you will, of muscle contraction in the muscle spindle organ. So let's talk a little bit about these muscle spindle organs. How many of them do you have per muscle? Depends upon the fine degree of control required by the muscle. So your hand, for example, contains a lot of these per gram of muscle, up to 120. Your calf muscle, your gastrocnemius, is not known for its fine control. It's known more for its power. That has 5 spindles per gram of muscle. So it's going to depend on how much fine control you have on that muscle. So if we look at a muscle, here it's attached to the bone with a tendon. Here we have our muscle spindle. We could see that it's innervated by a motor neuron and sensory neuron. And we're going to blow this up. The next slide is going to show you a blow up of that little square. It's a cartoon. Here's what it looks like inside that muscle spindle organ. First thing you should notice, the muscle spindle is separated from the muscle itself, the extrafusal fibers, by a connective tissue sheath. So whatever goes on here doesn't go on in here. This is separated from these structures. Now you have 2 different types of cells inside the muscle spindle fiber. You have these particular cells here where you contract at the ends, but not the center. These particular fibers have their nuclei in the center, and they're all lined up in a nice linear fashion, kind of like peas in a pod. And this is called a nuclear chain fiber. A nuclear chain fiber. You then have another type of cell, also has its nuclei in the center, but the nuclei are all bunched up like a bag of marbles. And this is called your nuclear bag fiber. nuclear bag fiber. So we have peas in a pod and bag of marbles. Nuclear bag fiber, think bag of marbles. Nuclear chain fiber, like links in a chain, they're all nice in a row. Now, your nuclear chain fibers are innervated by a motor neuron, but it's a different motor neuron than the one that innervates the extrafusal fibers. This motor neuron is called a gamma motor neuron. Gamma motor neuron. Good citizen, clean up your spills. Okay, when when the alpha motor neuron is activated, the gamma motor neuron is simultaneously activated. So whatever tension is created here by the activation of these extrafusal fibers, that same tension is also created here in these intrafusal fibers here. Now you have two types of sensory fibers that innervate the muscle spindle fiber. You have so-called flower spray endings that go to the ends. These innervate the nuclear chain fibers. These are so-called secondary fibers. The nuclear bag fibers have their sensory nerve endings wrapping around the middle of it. And that's your primary fiber. These are called annulospiral termini. Spiral endings, annulospiral termini. So the central region of intrafusal fibers don't have any contractile elements. All the contractile elements are at the ends. And the arrangement is either linear or random. They're linear in the nuclear chain fiber, random in the nuclear bag fibers. And your intrafusal fibers are one or the other. As you can see here, nuclear chain fibers, nuclear bag fibers. And then you have these two types of sensory fibers innervating the muscle spindle. Now notice, you don't have sensory fibers innervating the extrafusal fibers. Instead, any information about the muscle comes from the muscle spindle fibers. So as we said, annulospiral wraps around the center of the nuclear bag fibers. Flower sprays, secondary endings, they are located at the poles of the nuclear chain fibers, just like you see here in this picture. All right, here's how this works. If you stretch the muscle, you activate both the primary and the sensory endings. And this is going to tell the sensory nervous system about the length of the muscle. So you get impulses generated as a result of that stretch, producing both primary and secondary nerve endings. And the frequency of impulses will be proportional to the length of the muscle. Greater the stretch, the greater the amount of information, the greater the frequency of impulses that go to the sensory nervous system. Now, the secondary endings, the flower spray endings, they respond in a more sustained fashion, a tonic fashion. And if you have a sudden or very rapid stretch, that's going to stimulate both of these sensory endings. And it's a more powerful stimulus for the muscle spindle fibers. You'll get a tremendous amount of input from those, and you will probably then get a compensatory reflex jerk back. If, however, you stretch more slowly, more gradually, that has less of an effect on the primary sensory ending. And you'll have more, the input then will just come from the secondary flower spray endings, not the primary annular spiral endings. So if you activate them both with a really powerful stretch, you will get a very powerful reflex contraction. That reflex contraction can sometimes injure you. This is why your coach always told you to stretch slowly, right? You don't just, right down, you do it slowly, gradually, because you're going to stimulate the muscle spindle fiber just a little bit each time. You won't get this reflex jerk that can injure you sometimes and cause muscle spasms because of the way your muscle spindle fiber works. The fact that the secondary is going to work more in a tonic fashion, the primary won't be as activated by slow and steady sort of stretching than the secondary. Rapid stretching is going to activate both primary, secondary, very, very powerfully, and you'll get a reflex contraction that is very painful. You'll get a spasm. So that's why a stretch also will stimulate the muscle tendon organ and the Golgi tendon organ. The Golgi tendon organ is actually going to promote relaxation of the muscle. That's also why you stretch slowly. So here's why your athletic director always told you just to stretch slowly. Very good. So your gamma motor neurons innervate the muscle spindles. Your alpha motor neurons innervate the extrafusal fibers. The alpha motor neurons are thicker in their myelination, so they're faster conducting than the gamma motor neurons. Only stimulation of the alpha motor neurons will give you enough oomph to contract the muscle. Stimulation of the gamma motor neuron is solely for the purpose of the sensory nervous system, so that the sensory nervous system has a readout as to how powerfully the muscle is contracting. So with activation of the gamma motor neurons, you get isometric contraction of the spindle. And since contraction of the spindle, which is devoid of contractile elements in the center and towards the very end, the spindle is going to help you fine-tune voluntary control of the skeletal muscles and their movements. This was, I think, made plain to me once by actually a Little League coach I had. There were kids who, when the ball was coming to them, would get the bat down like this because like they're getting ready to really clobber the thing. And when you try to really clobber the thing, you don't have control. Also, when you clobber the thing, you have a tendency to overstretch, and you'll injure yourself more often than if you actually do a more controlled swing that actually has better technique. You'll also probably drive the ball a lot further than if you try, as the coach would say, if you try to kill it. That was his way of putting it. But you could always tell, because these guys would not hold the bat like this and cock like this. They'd always put the bat down like this, as though they were trying to, you know, hammer something to death. And I saw a couple of guys in my high school who didn't listen to the coach and did this sort of thing, getting ready to clobber the thing. And they had some pretty mighty shoulder sprains a couple of times. And that's simply because they overstretched. They weren't swinging in a way, essentially, that allowed their muscle spindle apparatus to give them the most information and balance, essentially, the swing, so you don't swing and then end up on the ground when you're done swinging. So the activation of both the alpha and the gamma motor neuron, that is called co-activation. Co-activation. And co-activation essentially dictates that that muscle spindle apparatus will experience the same amount of tension as the extrafusal fibers of the muscle, and therefore will provide very accurate information to the sensory nervous system about what's going on with the muscle. And muscle spindle fibers also responsible for reflexes. So reflex contractions occur in response to sensory input and sensory input only. They don't require any activation of the upper motor neurons. It's wholly through sensory information from the muscle, namely the muscle spindle apparatus and the Golgi tendon, and reflex arcs within the spinal cord. Reflex arcs take lots of shapes and sizes. So let's look at the simplest one, where you have a muscle stretch reflex. This is the one you guys tested when you tapped the patellar tendon in the lab that you did on reflexes. Remember that? And your foot hopefully kicked out. Yes? Some of you, your foot probably really kicked out. Some of your foot probably kicked out just a little bit, but it kicked out all the same. Then you did another stretch reflex with the Achilles tendon. You tapped the Achilles tendon. Hopefully you tapped it. You didn't brutalize it. There's no need for that. And the foot probably did something like that. Yes? Did you see that? Some of you are looking at me like I didn't do that. No? More or less, you tried it. Okay. So these types of reflexes usually are due to something called a monosynaptic reflex. And the knee-jerk reflex is a good example of a monosynaptic reflex because you don't have an interneuron. Instead, you have a direct connection between the muscle spindle and the motor neuron. So with this case, you have a dramatic contraction of the extensor muscles in the limbs in order to shorten the patellar ligament. And in this case, the muscle spindle detects a stretch in the muscle. It detects a very fast stretch. That sensory neuron from the muscle spindle then will connect directly to the motor neurons that activate the major quadricep muscles of the upper leg So the quad, it's called quad because it includes the four main muscles of the legs, the vastus internus, the vastus externus, the vastus medialis, and the rectus femoris. Yeah, rectus femoris. And those contract essentially. The sensory neuron will give a EPSP to the motor neuron. The motor neuron then activates contraction. It contracts, that shortens the patellar ligament. You now no longer get that stretch in the muscle. This is monosynaptic. Monosynaptic. And this is an example of isotonic feedback. So you tap the patellar ligament, the muscle spindle fiber detects stretch. You have a secondary fiber from the muscle spindle in the quadricep that then synapse with the alpha motor neuron of the quadricep. This provides a EPSP to the motor neuron. This then sends a signal. You get acetylcholine released at the muscle, and that's your knee-jerk reflex. That shortening then will cause the knee to kick out. And this is the most useful test if someone has degenerative disc disease. Because if someone has degenerative disc disease and they have a bulging disc, they will not have a knee-jerk reflex. And they'll have decreased strength as well. And with that, you cup your hands, you have them pushed down. And if they can't push your hands down, that's decreased strength. They Can you hear me now? Yes or no?
Yes.
When I walk away from the
Yes.
computer, then you won't hear me.
Okay.
I don't know what happened with the microphone. It just quit. No idea what happened. The light's still on. It's charged. I don't know. Now, the interneuron to which the Golgi tendon organ synapses will give the motor neuron to which it synapses an IPSP, an inhibitory postsynaptic potential. So it will inhibit muscle contraction and actually cause the muscle to relax. That's why stretching can promote muscle relaxation. And you can see that here. So this is a disynaptic reflex arc. Disynaptic reflex arc. You sense stretch on the tendon. You synapse with an interneuron. The interneuron is activated, but the interneuron then provides an IPSP signal. It hyperpolarizes the motor neuron, and therefore you get muscle relaxation because the alpha motor neuron is inhibited. Many reflex arcs also have reciprocal innervation. Let me demonstrate for you reciprocal innervation. Everybody, pick up your water bottle if you have one and bend it towards you. Now, in order to bend it towards you, what muscles did you have to contract? Flexors, your muscle flexors. What are your muscle flexors? Quick review of A&P. Oh, Jacob and John, come on, you guys should know this. In your sleep. Brachialis, and the biceps. So the biceps is a pronator, is also flexor. So the brachialis and the biceps. So if you're tensing your flexor, what do you have to relax as well? Your extensors. What's the main extensor of the arm? Tricep. So your reflex of the bicep is going to also include concomitant relaxation of the tricep. Therefore, many of your arcs will show reciprocal innervation. You get activation of the agonist muscle and inhibition of the antagonist muscle, which is shown here. It turns out the knee-jerk reflex consists of activation of the agonist muscle, which is the quadricep, but you also have a dual connection to an interneuron that provides an IPSP to the antagonist muscles, which in this case would be the hamstring, the semimembranosus, the semitendinosus, so on and so forth. Those will be relaxed at the same time that the quadricep will be activated. This is an example of reciprocal innervation. Reciprocal innervation. You activate the agonist muscle, you inhibit the antagonist muscle. So you do have cases of monosynaptic stretch mechanisms, but most of your reflexes are going to be your reciprocal innervation. And so whenever your antagonist muscles are stimulated, the alpha and gamma motor neurons that stimulate the antagonistic muscles will be inhibited. That's your rule for reciprocal innervation. Now, what about cases where you have to include two limbs? So for example, let's say John over here is playing third base. And let's say somebody, oh, who do we hate really? Mount Vernon, we hate Mount Vernon, right? Let's say somebody from Mount Vernon is sliding spikes up. You've got one foot on the bag, you got another foot in the way, you're gonna get a foot full of spikes. And you don't want that because that hurts. So you're gonna lift that foot while you catch the ball. Now, when you lift that foot, what do you have to do with this other leg? You have to extend it, don't you, to brace it. What happens if you retract this one and retract this one too? You're gonna go down. Then you won't catch the ball, you can't tag third base, and then this jerk won't be out. And that jerk, you want that jerk out, don't you? So some of these reflexes, therefore, have to cross to two different limbs. What they tell one limb to do, they have to tell the other limb to do the opposite. So this is an example of dual or double reciprocal innervation. Double reciprocal innervation where you have contralateral reflexes. So with cross extensor reflexes, say you step on a tack, say with your right foot, you're going to lift, therefore as a reflex, the right foot. That reflex will also include an extension of the left foot in order to brace the left foot so that you can raise the right foot. So when you raise the right foot, you are relaxing the extensors and contracting the flexors. But you're doing just the opposite on the left foot. You're activating the extensors and relaxing the flexors. That's cross extensor reflexes. So you step on a tack. Now you get that sensory information. That sensory information provides an EPSP to the flexor, but it also gives an IPSP to the extensors. Therefore, you flex the foot and get off the tack. You then have a branch of that sensory neuron that then innervates with a commissural interneuron. It's commissural because it crosses the midline. This will provide an EPSP to the extensors of the contralateral foot, and this will innervate with another interneuron that will provide an IPSP to the flexors of the contralateral leg so that you brace that leg properly. That's an example of a cross extensor reflex. Notice this does not work without interneurons because you need different results from these different alpha motor neurons. Those different results will be mediated by these different interneurons. Some will provide EPSPs, some will provide IPSPs. So this commissural interneuron provides EPSPs all the way around. But this interneuron here, that will provide the signal to the flexors of the contralateral side, that provides IPSPs. And Corinne, you're giving me I'm really lost look.
No, I'm just.
You're just chilling? When you look up at the ceiling, sometimes that means you're lost. You're just chilling. Okay. If I could put this on a quiz, you could answer it? Sure? All right. So the pyramidal motor neurons, upper motor neurons in the precentral gyrus of the brain, they're going to cross over in the pyramids. That's why they're pyramidal motor neurons. In the medulla. So they will include the lateral and the ventral corticospinal tracts. Neurons in other portions of the brain are going to regulate that motion. So the upper motor neurons are responsible for the signal to contract, the signal to move. However, refining that movement is a result essentially of these other tracks. And these are the extrapyramidal tracks. The major one being the reticulospinal tract. That originates in the reticular formation of the brainstem, the medulla and pons, and it inhibits lower motor neurons as previously described. And then the cerebellum gets into the action 2. It will provide the cerebrocerebellar tracts, and it is going to provide information from vision, hearing, and the vestigial nuclei that will provide information about balance. The cerebellum is going to connect to other nuclei. It connects to the vestibular nuclei, the red nucleus, and the basal nuclei. Virtually all your output from the cerebellum is inhibitory. And then these structures here will contribute to regulation of movement through the vestibulospinal, cerebrospinal, and reticulospinal tracts. These are involved with eliminating inappropriate neural activity, and the cerebellum is going to also provide the ability to coordinate your spatial judgment. So let's say Corinne's cheer team, let's say they lift Corinne up on one of these lift things, and Corinne's at the top. She's got her arms above her head. She is standing, I'm guessing, on somebody's shoulders. Sure, yeah, okay. She now has to adjust as that person adjusts, because that person is probably adjusting people under them. Corinne now has to sense that, and she has to adjust accordingly. That is all essentially the result of the vestibulospinal tract and also other tracts as well. What happens if that person starts to sway? Corinne is going to have to adjust her standing on that shoulders accordingly, otherwise she's going to take a tumble. That would be bad, right? Yeah, so she is going to adjust accordingly, and then when it's time for her to jump down, what do they normally do if it's time to jump down? They grab your legs and you take a leap or something? Or... Yeah. So when she leaps, is she going to do head first? Gosh, I hope not. She's going to probably, I'm guessing, kick her legs up like that and have them catch her, roll up on the ball, and they catch you like that because that's easier to catch somebody. Yeah. So again, she's doing that essentially based on information coming from these tracks here. We talked about the basal nuclei before, but they're going to act through the cerebrospinal tract and then also indirectly through the reticular tract as well. And they exert inhibitory influence on motor neurons as well, and they eliminate inappropriate motion as well, both at rest and also during motion. These are some of the results of damage to some of these upper motor neuron tracts. So if you have damage to the pyramidal tracts, the upper motor neuron tracts, you will have flaccid paralysis. The muscle is simply incapable of contracting. If, however, you have damage to the extrapyramidal tracts, And you have a variety of plegias. Plegia refers to paralysis, quadriplegia, paraplegia, hemiplegia, so on and so forth. That's due to direct damage to the spinal cord. Damage to the extrapyramidal tracts, though, will cause uncontrolled contractions of various muscle groups, causing things like chorea, things like this and things like that. St. Vitus dance, it's sometimes called resting tremors. That's a characteristic of Parkinson's disease. Intention tremor, that's due to damage from the cerebellum. And then things called Babinski's reflex. That's if you stroke the bottom of the foot. Normally your toes should do this. If, however, the toes are splayed out like that, that's what you expect to see in a baby. If you have damage to the extrapyramidal tracts, the toes will splay out. That's essentially you're recapturing the Babinski reflex when you go back to those more primitive reflexes. So some of you probably didn't have a good Babinski reflex. Those of you who are runners, the foot tends to get, there's a dead zone in the foot. So your foot kind of goes, yeah, what are you trying to do? Tickle me? I hardly feel that. The rest of you, though, your toes probably did something like this. Hopefully none of you did something like that. So the toes should just kind of do this. And that's due to just a stroke like that. And that sometimes the movement is very subtle. It's easy to miss. But other times it's very stark. Okay. The quiz on Friday will be over I guess it's chapter 12, is it? Yeah, chapter 12. All right. Yeah.
You said chapter 12 is going to be the questions about it.
Yeah. All right, let's get this there. Okay. Rats. There. Oh, stop that. Okay. All righty, good. Okay, guys, that moving? The slide's
It's not
moving?
moving.
Not moving. Moving, yes, no?
I still don't see it moving.
Still not moving. All right, one more time. Moving?
I can see your mouse moving, but the slides aren't moving.
about Jacob, but like I have the slides printed out in front of me, so it's not the biggest deal for me if the slides work or not.
I have them too, so you can just keep going and I'll follow along with mine.
Okay. Circulatory system. So how do you get things from one end of your body to the other end of your body? How do you get oxygen? How do you carry hormones? How do you carry nutrients? How do you get nutrients from your stomach to your big toe? It's through the circulatory system. So all the transportation that occurs in your body, it occurs via the circulatory system. The circulatory system also serves to regulate temperature. Sometimes surrounding things with blood vessels can also cushion things, so it plays a role also in protection. It also plays a role in the immune system as well. So the circulatory system consists of the heart and blood vessels. That's the cardiovascular system. And then the lymphatic system, which are the lymphatic vessels, the lymph nodes, the thymus, and the spleen. So the heart is a pump. It starts pumping day 22 of development, and it doesn't stop till the day you die. You ask me, that's pretty incredible. Doesn't take a break, doesn't take a break, doesn't ask for a raise, just keeps on going like the ever-ready bunny, except it's much less irritating than the ever-ready bunny. So at rest, it pumps about your whole blood volume per minute. At exercise, it pumps it a whole lot faster than that. The heart pumps the blood into a network of little tubes called blood vessels. These connect to the heart. They circulate from the peripheral tissues to the heart, to the lungs, and all the way back, and so on and so forth. Arteries carry blood from the heart to the tissues, veins return it back to the heart. Let's talk arteries first. Arteries are big, they're muscular. They will branch into arterioles. Arterioles are smaller, but they're still quite muscular. If you look at a cross-section of an artery and a vein, both will have smooth muscle, but the arteries have a lot more. The arteries also will have more connective tissue. They're just thicker, bigger vessels usually. From the arterioles, you go to so-called metarterioles to capillaries. The capillaries are 1 cell thick, and they are microscopic. They will feed the tissue beds. From the tissue beds, the capillaries will fuse into venules. Then the venules will fuse into veins, which then go back to the heart. All right, our time is up. And then we'll start here on Friday after the quiz, and so on and so forth.